all these people dick sucking the Trump administration are getting Barrett's cancer. They're getting throat cancer from dick sucking the Trump administration. But actually, the paper we're looking at today is talking about acid reflux and how it makes all your cells confused and a little delulu. So delulu that they start to accumulate extrachromosomal DNA and become cancerous. Let's take a look.
Twin Peaks. You know it's time to get the lab coat on. Twin Peaks. Yeah. Cause we out here being hella wrong. Twin Peaks. And you know we're never stopping till the hammer drop.
Plasmids are real. Like Bioshock.
Plasmids?
Mm-hmm.
No, I thought this was about cancer and tumor and extra chromosomal DNA.
Mm-hmm.
Yeah. How does that equate to plasmids? Well, Dispell, welcome back. Thank you for joining me on Beak to Beak today.
We've got a treat. We're back to the hard knock science, not the soft bullshit, these Substacks, these third-rate hack journalist articles. We're going to the premium stuff right here. And the forefront of technology and cancer research, extrachromosomal DNA. And it's not exclusive to Barrett's esophagus, which we'll learn about in just a moment. But we see extrachromosomal DNA in all sorts of tumors. And what we've got here is actually a case study on 2 consortiums of patients that were recruited to be studied in essentially their progression to cancer. Some will get cancer, some will not, and then you retrospectively take a look at them. Salty Squid, you have angels floating above me. What is going on? What the fuck are these symbols?
That would be an atom.
An atom? It looks like an angel.
Yeah.
Why have you summoned? What is going on here? Are these the angels of Techno Capital? This one is Techno and that one is Capital?
Yes, these are the symbols that are gonna change the world. They're hieroglyphics that will bring about the future of humanity as we merge with the techno-feudal crypto lords.
I'm so excited for this article that we've got this week because we are talking about extrachromosomal DNA in the development of cancer. The article is specifically in a form of esophageal cancer. But we're seeing extrachromosomal, yes, esophageal, congeal. We're seeing extrachromosomal DNA pop up in a lot of circumstances of cancer and generally genomic instability. We've got 2 articles essentially. We've got a cover brief, Tumor Biology by David H. Wang from News and Views, and we've got the associated full paper. Not by David Wang, actually by different people. I thought it would be like a member of the lab or something would have like written this, but nope, not at all. All right, what's your familiarity with DNA replication and cancer progression?
Well, I was about to say, should we just explain like basically how cancer forms for the people that don't know? I'm sure most people know, but like the reason cancer forms is because it bypasses the natural cell death cycle and goes beyond that, and it's basically an infinite cell that can live outside of the necessity to rely on other cells, but it feeds off them. Sometimes.
Yeah, they sort of like stop losing their like you identity. So like if Salty Squid got a tumor, those tumor cells wouldn't really be Salty Squid anymore because they're not exactly cooperating with the rest of his body. And part of that involves mutations, genomic translocations, generally genomic instability, and I guess replications of segments of the DNA even is what we're finding in this paper.
Yeah, I mean, there's lots of different ways that your DNA replication can get disrupted, and there's lots of sources of cancers and different kinds of cancers, but that's the baseline of it, is that it's cells that are growing beyond their natural cell death point.
Yes. And there are a couple like different qualifiers for what makes something cancer, because cancer, not cancer, that's a binary. But the real biological and material reality of the situation is that there is sort of a spectrum here. And with the paper that we've got today presents a case for analyzing the transition away from healthy to cancerous biology, specifically by analyzing the extrachromosomal DNA, which seem to be enriched for genes that carry um, oncogenes. Essentially, things that will make the cells live longer, be harder to kill, proliferate faster, evade the immune system more, and be more of a massive pain in the ass, or I guess a pain in the throat in the case of our article today. So we've got two today. Extrachromosomal DNA appears before cancer,
Twofer.
and extrachromosomal DNA and the cancerous transformation of Barrett's esophagus. Barrett's esophagus is not actually cancerous. The problem with Barrett's esophagus is with hyperreflux. So when you start to get older and your muscles don't work like they used to, and you're still eating the shitty high glucose corn syrup, processed suburban Walmart ass food, it's going to inflame your stomach, and it's going to make the acid rise up into your esophagus, where you get acid reflux, heartburn, all that sort of thing. But the problem is that the acid changes the environment of the epithelial cells in your esophagus, making them think that there's someone else. Your cells get a cell identity dysphoria because they're
Because they're
coated in so much
bathed in
acid.
acid.
Yes, exactly.
Beautiful.
And so the what this paper is presenting is they're saying we get this initial context of genomic instability where these cells are sort of in this transitionary state where some of them think that they want to be esophageal cells, some of them want to be more like stomach cells with goblet cells and other sort of secreting cells. And then that causes a context where they're sort of in this metastatable genomic state where they can accumulate mutations. And this paper identifies that TP53 is one of the major culprits. Uh, that's not big news. TP53, or sorry, yes, TP53, big culprit in cancer for quite a while, regulating the duplication of chromatin.
I was gonna ask Dispel before we get too deep into this. What is your clinical experience with cancer? Have you done any research into cancer? Because I've had quite a bit of experience with cancer, especially recently.
So yeah, I mean you're like really a hands-on sort of cancer guy. I'm more of like emotionally cancerous, like I would say personally cancerous, maybe like charismatically cancerous. That's sort of the extent of my
Yeah, I'm
work with
feeling the
cancer, I would
tumors
say.
growing every day. Every time we shoot a episode of Twin Peaks, I can feel a new tumor grow. That's
Well,
true.
I certainly might be partaking in a couple of those tumors because, well, actually, it is relieving because these are not delusions. You actually see the techno capital angels before us, so it isn't actually a delusion. They're actually there.
But
You're just
yeah,
going insane.
as I was gonna say, my experience with cancer, as you probably know, I used to work in veterinary oncology. So for a while there, I was doing chemo and working with end of life for cats and dogs that were going through cancer. So I've had quite a bit of experience, at least in veterinary medicine with cancer. I can't say I have much human experience with cancer, but that's why I was curious what your experience was with this.
Is dog cancer any different than human cancer? I don't know.
They get
Horse
different kinds
cancer?
of cancer. They're not exactly the same, but yeah, I mean, yeah,
Yeah, I would
they get
assume like
lymphoma.
the same principles apply. Yeah, this.
They get
Okay, yeah.
hemangiosarcomas, which humans do. It's not exactly the
Yeah.
same, and the levels of chemo that we give them are way, way lower than what we would give a human. And the goal is not to cure the cancer or put them in full remission. Like you're just trying to give them 6 months to a year and a half. I mean, sometimes they end up living longer than that, like 3 years or something. That's the best case scenario. But it's not like
And
human
squeeze these like
chemotherapy.
subsidized American farmers out of like the American tax dollars that flow into their farms so that they can keep their sick, cancer-ridden dog alive for another year. Wow, this is your experience with cancer. Yippee.
I mean, in my experience, the people that are paying to give chemotherapy to their animals are not the farmer guy that has a yard dog. It's the really bougie rich people that don't want Fluffy to die right now.
No, Fluffy,
Fluffy,
no!
no!
Whatever would I do? My 6-year-old would be depressed for all of a week and a half if my dog died.
I mean, personally, don't take my advice on this. This is just for myself. If my dog was fucking 13 years old and got cancer, I don't think I would put him through chemotherapy after seeing it. Like, I would just be like, you know what? You've lived a great life, and we're gonna have some extra burgers.
It's crazy you say that. It's because it's like, you make your dog high as a kite, make them shed every living, highly replicating cell in their body with chemotherapy. And you guys go, it'll be all right, little guy, it'll be all right, everything will be fine.
I mean, most of the time they tolerate it pretty well, but it can decrease their quality of life if they start having like really bad gastrointestinal disease.
Yeah. Well, gastrointestinal tract, very on track with what we're looking at today. I'm going to take the first and second paragraph. How does that sound?
Okay.
Okay. Yeah.
Yeah.
That sounds good.
Go ahead and read it.
Cancer, am I right? This is serious. Millions of dollars of perfectly productive humans go to waste every year because they develop cancer. This is a big problem, guys. Come on. Let's be
Yeah,
serious.
but Dispell, haven't you heard the theory that if they cured cancer, then they wouldn't be able to make money off of cancer? Come on, Dispell.
Yep. Okay, but like you cure one cancer, you're just gonna get another one. It's actually like a money-making machine. It is the money-making substrate. Sorry, cancer is the money-making substrate. You're not gonna get around it.
That and dialysis.
Oh, true. Okay. All right. A type of circular DNA not found on chromosomes, termed extrachromosomal DNA, can aid cancer growth by harboring cancer-promoting genes, or oncogenes. The unique structure of extrachromosomal DNA and its associated protein complex, called chromatin, boosts the efficiency of its transcription and the inheritance pattern of this DNA, resulting in many copies in a single cell, supports rapid amplification of the oncogene content and tumor evolution. Although present in many types of human cancer, extrachromosomal DNA was thought to be non-existent in normal tissues. On page 798, Lubeck et al., which is the attached paper we've got, I guess, later, report their finding of extrachromosomal DNA in non-cancerous esophageal tissue that is predisposed to cancer development and provide evidence that this DNA might help to drive non-cancerous tissue to become cancerous. So that's the metastable sort of uncertain cells.
Yeah.
Continue. Barrett's esophagus is a condition, we're explaining it, in which the cells lining the esophagus, figure 1, change their identity as a complication of the reflux of acidic contents from the stomach and intestinal bile back into the esophageal, and esophageal reflux. A minority of people with Barrett's esophagus go on to develop cancer after the cells have progressed through low-grade and then high-grade dysplasia.
That's what I was
Precancerous
thinking
states-
about.
Yeah, dysplasia is like a pre-cancer where
Yeah,
they've
it's
got
just
an
a
incredibly
proliferation of
unstable-
cells.
Yes, incredibly unstable genome, like under the microscope. And this is my biggest problem with this paper. No fucking histology. This paper, they talk about histology. They talk about, oh, we find extra chromosomal DNA. Where the fuck is it? Show me the fucking histology of these fucking cancer patients. Oh my god.
Yeah, go
Like,
off,
how can you
queen.
have a cancer study? No fucking histology. You fucking kidding me? It's crazy. Oh, I'm feeling stuffed.
You're such a histo bitch.
I do feel a little like stuffed up. I do feel a little like histamine action going on in my nasal cavity right now. Oh. All right, dysplasia. Not to be mistaken for my sister bird. Misplasia. Okay. Okay. These are precancerous states in which cells acquire an increasing number of genetic abnormalities and structural changes. So that this can actually be like a proliferating mass of precancerous cells, or almost like a silent, quietly mutating growth before it becomes really cancerous.
Yeah.
These are large-scale genetic abnormalities that can arise during dysplasia from mutations or loss of genes that function as tumor suppressors, such as TP53, and such mutations or loss can lead to whole genome duplication, genomic instability, or catastrophic chromosomal rearrangement called chromothripsis. No, chromothripsis. That's a new word. I learned a new word.
Chromothripsis. I love it.
What the fuck? You one-shot that? Are you kidding me?
I told you, I just had to
Chromothripsis.
dunk my donut brain in a little bit of coffee and we'd be fine.
Oh my god. Okay. New word for me. I discovered a new term. This is like insane. Basically, the hypothesis that like the human genome will just like at one moment just fracture apart, like the
Just
Holy Roman
fucking give
Empire.
up. The bridge will
Yeah,
break.
pretty much. It'll just give up into like these little like noodly bubbles, and then somehow it'll like put itself back together immediately afterwards, but like not in the same shape.
Life is beautiful.
Which can cause problems. It can cause problems.
Life is so fucking beautiful.
It is beautiful, but it also is sort of a sobering reminder that like we're facing an inevitable abyss of death and destruction and decay as our identity slowly dissipates into the environment around us, and our grip, physical and psychic, slowly loses its centralized meaning.
Well, yeah, you're just borrowing your atoms and you're gonna have to give those back one day.
I'm made of sunshine, is what you're saying.
Yeah.
Yes. So, TP53 was not the only mutation that they found in these cells. I think that's good. That's good for now. Do you have any comments from this this news and views paper from David H. Wang before we move on to the actual paper?
No, but I was just gonna mention this one figure is kind of interesting, and it's just going over the genome within your epithelial cells, which are the cells that line like your throat and your gut and like your really soft tissues in your interiors, and those epithelial cells are highly subject to being mutated and changed.
Yes.
They're not as resilient as
And-
other cells in your body are to resist this kind of mutation.
Yes. So once they get like a really catastrophic mutation like TP53, because once you get a mutation in TP3, it's kind of like you're kind of cooked. Like you are just going to get cancer. This is why a lot of young kids get cancer. It's like TP53 mutations. Gain of function, loss of function, like their cells are not splitting properly anymore.
Turning Point 53? Is that a new Turning Point USA organization that gives kids cancer?
There's no way you can't. I can't. All right, I actually have to know what TP53 means now, because like you can't get away with some stupid ass bullshit like that, and me not be able to actually you. Tumor suppressor transcription factor, of course.
I made you look it up.
Wait,
You
what is
fucking
the,
idiot.
what is the wait? What does the TP even stand for?
Turning Point, bro. I'm
Oh,
telling
tumor.
you.
No, it's fucking tumor protein. Are you fucking kidding me? It's tumor protein 53. I fucking hate how researchers name shit. Are you fucking kidding me? How the fuck are you gonna name an entire lineage of like human developmental factors after Sonic Hedgehog, and fucking one of the most like crucial genes to determine cancer affinity is just tumor protein number 53?
I don't know. I'm
my
actually in
God.
favor of us, going the way of not naming animal species after ourselves anymore and stopping taking the fucking credit for that kind of shit. Just name it something normal, add to the taxonomic tree, and then get out of the way.
What are your thoughts on abolishing species as a concept? Like we just stop thinking about species.
What?
It's a tough one. What if we just like stop thinking about species? Because
Can we
like,
start with race? Because that one's pretty shitty.
whoa. Uh, y- See, but like, race is like cognitively maintained, much like species is,
So you're
and
trying
there's
to
like a
say
similar
that you and
biological
I
rod
are the
underneath.
same species? We're the same thing?
Well, because like the classic, like simple definition for a species is whether or not two creatures can make viable offspring. And
Kind of.
the thing is that there are a lot of species that can make viable offspring. It's just that their like mating
Well,
periods
it's not just
are
that their
like
offspring
shifted
are
by
viable,
environmental
it's
factors.
that their offspring can then go on to make other offspring, which most of the time it
Yes,
doesn't.
that's
It's a
that's
dead
viable.
end.
That's viable.
Okay.
That's viable. That's viable. That is viable, right? Okay, we've got we got Mr. Marketable on the viable case right now.
I mean, it could mean also a viable embryo that will, be born.
Oh yeah, produce a thing. Yeah, I was okay. I think the specification, yes, very important. The viability is the next generation works, and then the next generation also works as well, because then you get like stuff like mules,
Yeah.
which are great. We love
Or
mules.
ligers.
Not, huh?
A liger?
What?
But a liger and a liger can't fucking make another liger. It has to be a tiger and a lion.
A liger is what you went for. Okay, all right.
Brother, have you not seen
Interesting.
Napoleon Dynamite?
No.
No?
You're like 50, you're literally 50 years older than me.
Oh
Like you
my
were literally
God.
born. You were born watching FDR on
I'm
the
2,000
television.
years older than you, okay? Get it straight. I'm a kaiju.
Your
I've been here
evolutionary
forever.
history is
I was
probably
there
much
when
older than
Jesus
mine.
was on the cross. I
200
witnessed it
is
happen.
generous. 2 million years maybe, cephalopod. Billion maybe even. Who knows?
Speaking of billion, did you hear that Elon Musk became a trillionaire? We failed, bro. We lost as a species.
Did we lose as a species? Like, but like now that all the techno capital is centralized in a single ketamine-induced addled freak who has surrounded himself with a cadre of eunuchs specializing in various fields,
He has
looking to get their own bag.
$1 trillion. Let that sink in.
I don't. When is he gonna buy like a country?
When is he gonna revive comedy?
I
I've been missing
I
comedy.
I don't think it's gonna happen.
No, it's gone forever.
I don't think $1 trillion is going to revive comedy.
Are you sure?
It's not. It can't come from wealth, I don't think.
No, you're right. You have to punch up at wealth. You have to be the jester shitting on the king's lap.
You ever be on your 40-meter yacht in Monaco and you like texted your towel boy 30 minutes ago to bring you a new towel after you got into the shower and you forgot to bring a towel, but you wanted your favorite towel boy, you know, the little one with like the arms that you can like wrap around to get, you can get like your pinky and touch your pinky and your thumb to that towel boy, and he hasn't like come by yet. It's like, it's really brutal. That's what I imagine trillionaire stand-up is.
I don't think you should be talking about Umberto that way.
Who? Umberto? Oh my god, you cannot be making cannons for these people.
Why not?
Because we're taking a look at esophageal cancer development.
You don't want to go on a
DNA is what we're
tangent
looking at.
about millionaires' yachts and their Filipino slave boys?
I said that we can, and we will partake, and we will also mention that they will be harvesting the little Filipino boy for both of his kidneys and his bone marrow for his stem cells, so that we can use those stem cells to make little cancerous mini-me trillionaires.
How far off are we from them, like, growing clones to harvest organs from?
Pretty close.
I think we're super close. Like, 20 years?
5.
5?
5
Oh,
years.
fuck.
I would say 5 years proof of concept, 10 years industrial scale.
Would they even allow that to go industrial scale? I mean, they like to keep their secret shit
Please.
pretty nasty down in the sex dungeons on Epstein Island, where they're taking the blood of 20-year-olds to feed their vampiric needs to live forever.
I think I give it 10 years.
All right, we'll see.
But these extra chromosomal DNA. So if we go to
Oh,
the
you wanted to
main
talk about a
paper-
paper today? I'm sorry. My bad.
Yes, the article, the extra chromosomal DNA and the cancerous transformation of
Oh
Barrett's esophagus.
no!
Paper by Paul S. Mitchell's lab. Because these extracellular chromosomes are not just a product of genomic instability predisposing people to cancer. They could also be the future of plasmids. Just like Bioshock, you will be shooting lightning from your hands. Hit it, salty squid.
Oh, really? That sounds dope. The paper is called Extrachromosomal DNA in the Cancerous Transformation of Barrett's Esophagus. The abstract here says oncogene amplification on extrachromosomal DNA or ecDNA drives the evolution of tumors and their resistance to treatment and is associated with poor outcomes for patients with cancer. At present, it is unclear whether ecDNA is a later manifestation of genomic instability or whether it can be an early event in the transition from dysplasia to cancer. Here, to better understand the development of ecDNA, we analyzed whole genome sequences, WGSs, data from patients with esophageal adenocarcinoma, or EAC, or Barrett's esophagus. I think that's why I thought that they were equating the adenocarcinoma with Barrett's esophagus, but I guess it's not quite the same thing. The Barrett's esophagus happens when it starts to be dysplasic.
When you got heartburn. It's when you got to take your antacids.
But then it can later develop into an adenocarcinoma. So
Yes, again.
it continues. These data included 206 biopsies in Barrett's esophagus surveillance and EAC cohorts from Cambridge University. We also analyzed WGS and histology data from biopsies that were collected across multiple regions at 2 time points from 80 patients in a case control study at the Fred Hutchinson Cancer Center. In Cambridge cohorts, the frequency of ecDNA increased between Barrett's esophagus associated early stage 24% and late stage 43% EAC, suggested that ecDNA is formed during cancer progression. In the cohort from the Fred Hutchinson Cancer Center, 33% of patients who developed EAC had at least one esophageal biopsy with ecDNA before or at the diagnosis of EAC. In biopsies that were collected before cancer diagnosis, higher levels of ecDNA were present in samples from patients who later developed EAC than in the samples from those who did not. We found that ecDNAs contain diverse collections of oncogenes and immunomodulatory genes. Furthermore, ecDNAs showed increase in copy number and structural complexity at more advanced stages of the disease. Our findings show that ecDNA can develop early in the transition from high-grade dysplasia to cancer, and that ecDNAs progressively form and evolve under positive selection.
Yup. If my microphone could catch me snapping,
Yeah.
you would hear that right now. Because you were fire. It was so spot on.
Yeah, I mean,
That's the paper.
The thing that's interesting about this though is that can they then go on to use the detection of the you did that too, extrachromosomal DNA. Can they use the detection of that to use as like early cancer detection?
Yeah. That would be
Like
super
before
sick.
it even fucking forms, you're just noticing that things are becoming dysplasic, and then you notice there's some extra chromosomal DNA being formed. Then you start to intervene before the cancer tumor even can form.
It's crazy how humanity will just convene on, a couple of my homies do have a couple of extra chromies. And by chromies, I mean extra chromosomal chromies.
All my best homies have extra chromies.
But like, okay, in this circumstance, these homies with chromies are gonna give you cancer. So if you scroll down the article a little bit to the section labeled ecDNA and Barrett's esophagus, this is where the story starts. This is where they're investigating the sort of like earliest progenitor cancer cell in these populations, and we can get a gauge on whether or not this is a feasible technology for like diagnostics. ecDNA was not detected in any of the non-dysplastic Barrett's esophagus samples or any of the LGD samples in the cross-sectional Barrett's esophagus surveillance Cambridge cohort, which means no cancer, no ecDNA. Very strong start. Continuing. By contrast, ecDNA was found in tumors from 13 out of 51 patients with early stage EAC and in tumors from 38 out of 88 patients with late stage tumors. So, not all tumors have extrachromosomal DNA. Only about 40%. Senor Squid, you catch that? 40%
Yeah,
of cancers
that's
even
kind of a
end up
bummer.
having. Yeah, it is kind of a bummer, but that 1 in 4, that 25% of like pre-diagnostic potential, that's pretty good. That's pretty good. It's usable. The occurrence of ecDNA was significantly enriched in early stage EAC versus non-dysplastic Barrett's esophagus or LGD, with increased frequency of EC in late stage compared to early stage tumors. ecDNA was detected in a nearly identical fraction of an independent cohort of late-stage EEC tumors from the TCGA. Great stuff. This next paragraph, not too much interesting. Actually, hold on. There's. The FHCC study incorporated multi-regional, longitudinal sampling from before and after cancer diagnosis. We examined the development of ecDNA over time in biopsies from patients with Barrett's esophagus tissue that progressed to an EAC endpoint versus those patients with Barrett's esophagus tissue that remained benign at the highest detectable disease state. At cancer diagnosis, this is TP2. ecDNA was detected in samples from 11 out of 40 patients with a cancer outcome who developed EAC of 28%. Consistent with the 25 frequency of ecDNA that was found in the Cambridge cohort of patients with early stage cancer. EAC was detected in biopsies from only 1 of 40 patients with a non-cancer outcome. We're seeing a pretty dramatic, it could not be to random chance alone. There must be some explanatory power to these extrachromosomal DNA showing up in these cancer patients.
And how are they
They also
detecting
did
the
something else.
extra chromosomal DNA?
They are using whole genome sequencing, which means they grind up the tumors and just grab all the DNA that they can. Good question though, because they do have like specific kits that they use. They use the CNV kit and ASCAT with the Amplicon Suite pipeline, which was used to identify candidate seed regions for the detection and characterization of ecDNA using Amplicon Architect and Amplicon Classifier. And circular visualizations of candidate EC structures were generated using CycleViz. Hold on, where did they get their sequencing data? Whole genome sequencing by Illumina was performed with 50-fold coverage for the tumor and 30-fold coverage for the matched germline control.
So I think
Aberrant cell fraction and plenty were reported. Okay.
if you were gonna use this as some kind of like pre-diagnostic tool, because I'm thinking about like real world uses of this kind of information, like you would have to at least know that there's dysplasia happening first to even know to sample an area.
Yeah, so this is one of those things where it sort of just takes like voluntary surveillance,
Yeah.
where almost like mammograms, pretty much,
I
or
mean,
like vaccines.
men out there, you should be feeling up your testes and making sure there's no little friends or little growing on there. Women,
Guys,
feel up the mammaries. Make sure there's no little
that's,
tumies going in
I
there.
don't think, I, you need, I'm pretty sure like an MRI for that. But like men, it is okay to moan when your doctor is
You're
checking
thinking of a
for
mammogram?
for chromies in your prostate.
Chromies in
And
your
by chromies, I
prostate?
mean extrasomal chromosomes which could potentially rise to cancer. I don't know if
Yeah,
that's
do not let
something that's
chromies be in
prostate
your
cancer-ish.
prostate, but you can let
Yeah.
homies be in your prostate because it is Pride Month.
Yeah, pretty much. So Illumina sequencing, it's good stuff. It works well. 50x coverage, pretty great. 30x coverage, awesome stuff. And then it seems like they just grabbed a bunch of like little sections of DNA and C and V kit. I don't know how that would be able to. Oh, okay. So they detected the number of copy number alterations. And then they use the copy number alterations to predict ecDNA.
Oh, see, I see
So I
little
did look this
figures
up.
down here that are talking about the histology. You're upset that they didn't show us any histology.
I can't find any of the histology. It's like you want me to believe that the chromosome has shattered catastrophically and some of these tumor samples have coiled up particularly uh, evolutionary advantageous for cancer genes into little plasmid circles, and you can't even show me where they are. Like point on a fucking screen where they are. Don't just give me some statistical slop, and it doesn't exist in this paper.
That's
There are other
unfortunate.
papers. There are other papers which try to visualize it using a pretty cool technique called FISH, which is in situ hybridization, where you can visualize the production of RNA and get like a little fluorescent signal anytime it happens.
Oh,
And
that's
pretty much
cool.
all this extra chromosomal DNA looks like it's just like little dots throughout certain pockets of the DNA or the nucleus essentially. Oh, here's actually a pretty good image. Go to this very first image that they've got, Figure 1. Great visualization of these extracellular pieces of DNA. And the problematic thing is that mitochondria have their own DNA. So it's like you
They
kind of
do. Why?
already have extra chromosomal DNA.
Why are you
Oh
complaining?
man. Okay. So it looks like these extra chromosomal pieces of DNA, essentially they've got promoters and enhancer sequences on them so that they can amplify the expression of like their original gene. This is a visualization of extrachromosomal DNA. It's a schema. And these self-aggrandizing, accumulating little bastards will just form a circle jerk of enhancer and promoter regions with transcriptional machinery to just generate a fuckload of these cancer genes, making the cell even more and more hysterical and insane.
It's unfortunate. It's kind of beautiful though.
It is gorgeous. And it is like the weird thing about it is that that's how bacterial genomes work. They are circular. And it is kind of weird that like the human genome, we can have things put into it and then portions of it will like fall out and then form a circle. And potentially we could use this for good, potentially we could use this for evil, but for now we're using this to figure out what the fuck is going on. So if we go back to the paper of the week, my beloved squid. So the article, we talked about the extrachromosomal DNA in bear's esophagus. They found not much in the people who didn't get cancer. And the people who did get cancer, about 40 to 25% had extrachromosomal DNA inside them before they even detected the cancer. So we go past a couple of other things. Yeah, it's associated with the cancer. Yeah, it's associated with malignant transformation. Yeah, it's enriched with oncogenes. What happens next? Extrachromosomal DNAs with common origins. The authors say, to compare the fine structures of ecDNA across multiple time points in biopsies from the same individual, we developed an amplicon similarity score ranging from 0 to 1. Genomically overlapping ECs in different samples from the same patient showed a high similarity consistent with a common origin. All 10 genomically overlapping EC pairs from within the same individuals re-identified between different biopsies showed significant similarity. Thus, extrachromosomal DNAs detected in precancer are frequently maintained through the transition to cancer, and genomically overlapping extrachromosomal DNAs identified from multi-regional sampling are likely to have a common origin. Together, these DNA suggest that ecDNA can be a truncal event in the formation and evolution of esophageal cancer. So basically, cancer cells at different parts, so in figure 3, they've got first in number 1, surveillance endoscopy. This is no cancer, 60 years old, acid reflux, fine. Things are fine, okay? Then we go to time point 2. Somehow this is both. We've gone from 60 years to 62 years old at the very top, but somehow this transition is 5.6 years, I guess. Can you just get cancer all within like 5 years, I guess?
Yeah, absolutely.
Yeah. Yeah, you're 60 years old.
So
Okay.
it's in the
Basically.
esophagus picture, surveillance endoscopy, it's showing where you start at, and then 5.6 years later, if you're fucking getting all this bile constantly shot up into your esophagus, which I thought it was the proximal esophagus, but it looks like it's actually the distal one, which that makes way more sense. If
Closer
you
to
keep
the
getting,
stomach, yep.
yeah, if you keep getting acid shot up into there, then you have the chance here at figure number 3, 6.5 months later, you start to form dysplasia, and then that's when you start to get extra chromosomal cells, and that's when you have the risk of a tumor forming.
Correct. But take a look at this. At time point 2, we've got ecDNA accumulating at point 3, and then 6.5 months later, we find ecDNA has moved down, and it's been combined with ecDNA2 at the very top. And then at the very top, we find that the cancer cells, or this plastic cells, have accumulated both EC2 and EC1. So essentially they're collecting these extrachromosomal DNA, almost like Pokemon, in order to become more cancerous.
Gotta catch them all, bro.
Yeah, gotta catch all the dysplastic extrachromosomal DNA. So in subfigure C, they've got the actual EC1 and EC2.
If I train up my tumor cell really well, would it become something else? Will it evolve into a more stronger metastasizing tumor cell?
Yeah, probably. Yeah. Yeah. If you train it, yeah.
Then I could go down to the local St. Charles Hospital and fight other cancer patients with my tumor cells, and we can have Pokemon battles with our tumors.
That would be pretty cool, and it might be a way to describe how we treat cancer. I'm not. Yeah, kind of. Pretty much. Yeah,
But-
sure. I'll buy it.
Go ahead and continue. My shenanigans are satiated.
So they also make the case that not only are these extrachromosomal DNA collected like Pokemon, they're actually selected for, and they're selected for extrachromosomal DNA segments that promote the expression of cancer genes. So they say, selection and evolution of ecDNAs. We detected a marked increase in the frequency of ecDNA in biopsies from patients with a cancer outcome before clinical detection of cancer, and even higher levels of ecDNA in biopsies or resections from later cancer patients. To better understand these observations, we characterize 137 ecDNAs across all samples from 75 patients with ecDNA positive Barrett's esophagus, Barrett's esophagus derived HGD, or Barrett's esophagus adjacent EAC. So adjacent to the cancer, but not necessarily cancer. EAC DNA copy number was significantly higher in the cancer samples than in the pre-cancer samples. Although the lengths of genomic regions captured on ecDNA were not significantly different between cancer and pre-cancer, the complexity of the structural rearrangements in ecDNA-derived regions increased between pre-cancer and cancer, suggesting a significant increase in the heterogeneity of ecDNA structures with the evolution of tumors. Essentially, they gain more and different pieces of ecDNA Pokemon. We next investigated copy number changes in 8 pairs of clonal ecDNA in which the same ecDNAs appeared in different sequencing biopsies from the same patient, and for which the biopsies had a windowed histology data available, which they don't even show us. Supplemental data, table
You're
1
so
is
salty
what
about
they
that.
cite. It's literally just a table of the histology that they gatekeep. When both ecDNA occurrences were associated with the same histology, the ecDNA copy numbers were similarly high. However, if one sample was associated with more severe histological status than the other, the EC copy number was significantly higher in that sample. This data suggests that ecDNA confers a strong selective advantage to the Barrett's esophagus clones that eventually progress to cancer, and that precancer ecDNAs are subject to continued evolution during malignant transformation and progression, leading to increased heterogeneity and copy number. Let's take a look at figure 4.
I'm taking a look.
Let us meander around all the pretty dots and figures and lines that they've got,
There's
and
so
draw
many
our
pretty
lines
colors.
to this vertical cliff shaped one under G.
I see it. I see it.
So we're actually going to combine E and G. So E says that of the patients that had extrachromosomal DNA and got cancer, which was about 50 to 25%, 30% of those people had multiple copies of different ecDNA in their cancer. Stacking that bag, getting that bread up. Now, they don't really associate these with any sort of like invasiveness or tumoricity, which is a little disappointing. I guess the denizens of the ecDNA speak for themselves. And what do we find? In figure G, we've got the list of all the unique pieces of DNA that they find on these ecDNA. Big names like ERBB2, this is in breast cancer, EGFR, growth signaling, GATA6, transcription factor, Mike, massive sort of like whole genome overdrive, essentially. Like Mike, when you're expressing MYC, you are just creating more mRNA. You're just creating more protein. It is essentially putting your cell on steroids. MYC. Very, very big cancer-associated gene. We've got other cell checkpoint marks, CDK12. We've got GATA4, another transcription factor. All sorts of good stuff going up and down this list. Doesn't it excite you? Like, all these like different cancers have these different plasmids, and like essentially we can probably just like inject these little pieces of circular DNA into people, and maybe we can get them to express a little bit more protein that otherwise they might not have. But like, okay, sure, their genome will be a little bit more stable, but it's like better than the alternative, right?
Hey, if you think you can get us to the plasmid gods, I will follow you. You can be my savior.
I don't think so. I don't think so.
No, you're the bioengineer
I'm
here. I just stick my finger
This
in
is
puppies'
sci-fi.
butts.
This is not reality. When I say that plasmids are going to give you lightning, it's sci-fi.
Oh.
But like the original BioShock was kind of cool because of like its anti-accumulation, anti-insane futurist like planning for like a utopian free capitalism, that they also let you modify like your genomic information with plasmids.
I mean, I
Very
kind
cool.
of feel like Rapture is just like what Elon Musk is going to do with Mars Colony.
Yeah,
If he ever
sadly,
makes
but
it.
it will make great like Resident Evil games.
Oh, hell yeah. Like once the Mars Colony has fallen, then I'm going to go take a field trip there.
Okay, that's a great idea. Yeah, we'll go
Right?
together.
You want to go together? Oh my god, that sounds like such a good bro date.
Imagine like instead of vaccines, you just got like an extra chromosomal piece of DNA with all the promoter and enhancer sequences that you could need in order to promote or stage an infection. It's like I guess the boundaries are like endless. And what could go wrong introducing extra chromosomal DNA? what could go wrong? Because it's not like 100% of people with cancer have ecDNA. It's more like 50 to 25%. So giving somebody ecDNA isn't necessarily just going to give them cancer.
No, but isn't it true? I don't know if this is true, but doesn't it correlate how many different types of Pokemon you collect and how many distinct type of Pokemon you're collecting, the more likely that they are to evolve into a cancer?
Yeah, maybe. So like I guess like if we could get like a national profile for all the extrachromosomal DNA that you've grabbed, all you've collected, almost like you know what it should be. Tinder, Grinder, all of these sorts of apps, Hinge, they should be integrated with a national health system that tracks people's STDs and extrachromosomal DNAs and embryo selections. That's what we really need, Salty Squid. We need more visibility on the human genome for the future. I think it's critical.
I mean, we're doing our best out
The-
here. We're raising awareness.
The market demands it, actually. Maybe these pieces of a chromatin are like acting up because they don't feel included in the genome. And the important part would be, and the responsible choice would probably be, to reintroduce them into the chromatin. And so it's like, okay, so what if you've got a couple of copies of a certain gene? Like, genes duplicate all the time. What's one more? Like, what doesn't kill you makes you stronger, honestly.
I've already got plastics in my fucking blood. I might as well have extra chromosomes.
I 100% agree. Like, comment, subscribe for extra chromies.
All my homies with extra chromies, go down into the comment section and tell us what you're gonna do with your extra chromosomes this weekend.
Homies, like and subscribe. Chromies, well, we'll talk about that another time.
Chromies hit the bell.
I-
Homies like and subscribe, but chromies hit the bell.
That's a wrap. That's a wrap. That's a wrap. Holy shit.
Thank you for joining us on Twin Beaks. This has been Beak to Beak, where we've been talking about chromosomes and cancer and how you can avoid getting extra chromosomal cancer. Don't upset your stomach. Don't be eating Takis and Mountain Dew late at night while playing video games, because you will get Barrett's esophagus, and then you will get dysplasia, and then you will get cancer and die probably.
The propaganda I'm following for this summer, the acid reflux pipeline to esophageal cancer
The pipeline is your esophagus,
for acid reflux.
and the acid reflux is the propaganda.
Yes, it's caused by the propaganda. Cause it's so basic. Heyo.
Twin Peaks. You know it's time to get the lab coat on. Twin Peaks. Yeah. Cause we out here being hella wrong. Twin Peaks. And you know we're never stopping till the hammer drop.
For my friends, my family, for my well-being, can you, Mr. Marketable, carry this one for me?
I would if I had slideshow images. So unfortunately, I cannot carry you. But
You fucking
you're
have.